So dear student, today we will start the new genus Oedogonium, and it's a very important green algae. That's why both the theory part as well as the practical part will be discussed today. And I will explain in the end what you have to do, especially in practical. So Oedogonium, as you know that it's a member of green algae or the chlorophyceae. And these are the two classifications given by Fritz and Lee. There is no difference at all. Means if you will see that Fritz placed it in under division algae, and then class chlorophyceae, order Oedogonians, family Oedogoniaceae, genus Oedogonium. Apart from putting it under division Chlorophyta, rest of the things are almost same even in Lee classification. The justification of this classification will be discussed in the end. In this particular algae, we will justify up to genus level. So this is a photograph showing the luxuriant growth of Oedogonium, but it doesn't mean that all the algae are only Oedogonium. So Oedogonium's names had been derived from the Greek word oidios and gonos. Oidios means swelling and gonos means reproductive structure. So this name had been derived from this Greek word oidios and gonos. That's why it becomes Oedogonium. Please note it down these terms. That is a filamentous, unbranched, submerged freshwater alga. Okay. And you can assume, you can predict that yes, if it is a freshwater pond or lake where the water current is almost negligible, means when you will see the standing water or where the stream flow is very low, then you can assume yes, this is the area from where we can collect this Oedogonium. Easily identified by characteristic zinc at the apical end of the certain cells, which are known as cap cells. But for this easy identification, you should observe it under the microscope only. Okay, you cannot see those cap cells by naked eyes. These species may vary in dimension. If you want to study the different species, the basis of the speciation are mainly the structures of vegetative cells. Some species have more bigger cells than the others. The structure of oogonia. Okay, zygote wall ornamentation. Okay, what is the thickness, whether it's spiny or normal, and antheridia position. Okay, whether it will appear towards the apical end or towards the bottom end or in the center, or it's a macrandus type species or nanandus type species. So species can be divided on the basis of these structures. Although it's very rare, but there are some species which are edible in few tropical, subtropical areas. Algae can also form conspicuous bloom. I hope you know the meaning of bloom. Bloom का मतलब पता है आपको? Algal bloom. Yes. There you can see the luxuriant growth of algae. Okay. And this Oedogonium can also take place in the formation of that algal bloom. Okay. There is a difference in the young thallus and the mature thallus. Okay, and you will understood it once we will complete this lecture, why it is so. The young thallus is usually get attached, means it's always get attached with a hold fast to the plant. That's what is known as epiphytic. Or if it is not attached with a plant, it may get attached itself with any substratum like stone, wood, or like animals, dead animal parts or live animal body. So it may be epiphytic, it may be epizoic, it may be epilithic. Okay, depending upon the substratum, we can divide it like that. But once the thallus is fully mature, so it will start floating on the water surface. It gets detached from its substratum. So if you will see a mature filament, there are more chances that you will not be able to find out the holdfast. And there are some aquatic species, and the most important is Oedogonium elegans. This species is the most commonly found species, aquatic. However, there are some terrestrial species like Oedogonium terrestris, but these are very rare. Most of them are aquatic species. Before we start, we should know how to prepare a slide. So last week we prepared the slides in the laboratory, and this is the original material. Like this is one of the vial in our lab. Again, with the help of forceps, we should take only a small portion of Oedogonium. Okay, oh, that's very big. Now you can see how small it is. Okay, you can see that there is some filamentous thallus-like structure, but you cannot identify it by your neck, right? It may be anything. There are some other filamentous algae like this, like Spirogyri may be there, Ulothrix may be there, Cladophorite very common in this area. Now you will tell me what these are vial for. ये कौन सी क्या वायल है बताइए बताइए वे वायल्स जब आएंगी जो what are those vials for?
For staining.
For staining. What is that stain?
Sir, it's iodine and it's used for starch.
It's iodine and it is used for starch. What it will do with this starch?
Sir, it will bind to it.
It will bind to it, yes. And what it will form? I'm not asking the chemical reaction. I'm asking that what it will do with this. Yeah, like this is the iodine, and we will use it for staining purpose because it contains starch. But what happens when it will take the starch, react with the starch? Can you see it? क्या हुआ यहाँ पर?
Yes, sir.
It's become bluish black, okay, because of the presence of starch. Then you will take one more clean slide, mount it in the glycerine, and again you should take only very small portion of this. Whatever I have taken, that is also not required. Only that much is required. And the most important thing is you have to try to separate out all the filaments as much as possible without damaging the cells, because you have to use the needle over here. You cannot use brush for Oedogonium. And again with the same technique, with the help of a forceps or needle, gently cover your material with a cover slip. And if extra stain or mounting medium had been come out of the cover slip, you have to remove that. Then we will observe it under the microscope. So before showing its morphology under the microscope, you should know the plant body is a thallus showing apical basal polarity. Means I will show you. It is from top to bottom. The topmost cell is known as the apical cell, whereas the bottom cell is known as the holdfast cell. The thallus is long and branched. Whatever filament you will observe under the microscope, you will find out there is no branching pattern is there. Okay, it is made up of single row of elongated cell. Placed end to end means there is one cell, then next cell, then next cell. Okay, like this. मैं थोड़ा draw कर देता हूँ यहाँ पे. like one cell, you will see this kind of, I'm very bad in this. Okay, you will see the actual structure, but I'm just trying to explain what is happening over here. On the topmost, you will see the apical cell, a dome-shaped apical cell. Then all the cells you will see in the single filament, and in the bottom you will find a hold fast. Okay, the structure is different. इतनी गंदी नहीं होती जितनी मैंने बनाई है, and it's a long structure. That's why I use this dotted line over here. Okay, sometimes क्या होता है, कुछ books में होता है, they only draw a continuous line and only are the cells बना देंगे एक filament में, but it's not like that. There are more than 20, 40, 50 cells in a single filament. But it's not possible for you to draw all the cells. So what you can do, just show the apical portion, then few intercalary cells, and then the bottom hold fast. The next is filament that I have already explained, a club-shaped basal cell or hold fast, this one, dome-shaped apical cell, this cell, and intercalary cells. There is three types of cells, the topmost cells and basal cells, and all the cells are known as intercalary cells. Now we will see the morphology. This is what you will observe under the microscope. This is the same slide we are observing it, जो हमने बनाई थी. But still you can see that it's not a good slide. We were not able to remove or separate out all the filaments from each other. That's why they are appearing entangled with each other. And we are unable to see the actual structure of single cell. But it is just only for showing how it will look like under a microscope. So this is the hold fast, the bottom cell. Please just see it carefully. The filament is generally attached at the lower end by means of a basal cell or hold fast. This is the hold fast. ये structure को hold fast बोलते हैं. And hold fast is achlorophyllous. Sometimes students get confused that this is a hold fast cell. No, this is not a hold fast cell. This is the hold fast only. Okay. So this particular structure don't contain chloroplast, but this may contain. The next cell, this is a part of intercalary cell. Okay. In terrestrial species, the hold fast show rhizoid-like outgrowth. यहाँ पे आपको नहीं दिखाएगा देगा, but जो Oedogonium terrestriae, you may assume that rhizoid-like structure may also be there, appearing coming out from this hold fast. Okay. So see the beauty of nature. I will show you in the slide. So now you can see this is a hold fast. Now you can assume how long it is. We are just focusing on the bottom part. Can you see this structure? Yes or no?
Yes, sir. Yes, sir.
Okay. And you can also see there are some filament-like structure are coming out. It may be other filament, a very young filament of Oedogonium, or it may be a blue-green algae, or it may be something else. Okay. Or it may be a part of the tissue, जो animal से और plant से हमने इसको detach किया है. It may be part of that thallus also. Okay. But it seems that it's a very young Oedogonium. Okay. There will be more photographs, still photographs, and this. So this is one more photograph. Here you can see also that there are so many tissue-like structure when we try to detach it from the from that substratum. But here there is no something like structure like that. So it shows that those fiber-like structure are not the permanent structures. Here you can also see. This is one more photograph. This is one more. There are different, means depending upon the substratum जिससे attach होते हैं, you can see how it look like. It's just like a structure. ये किसे रिजेंबल कर रहा है आपका स्ट्रक्चर? आपने देखा है कभी स्ट्रक्चर ऐसा? Have you ever seen such kind of structure in your daily life?
Sir, vacuum cleaner.
Oh, sorry. That's the problem in PPT. The moment I click it, it will restart again. Sorry. So until unless we are coming to that point जहाँ पे हमने छोड़ा था, तो वैक्यूम क्लीनर में आपने देखा होगा, और यू हैव सीन दैट जो कारों के शीशे होते हैं, if you want to protect yourself from the sunlight, there is some net like structure, and in the center of that net like structure you can see a rubber section like of things. When you will fix it with your glass of your car, so it will get attached itself with the car window. Am I right? मेरे पास है नहीं, अगर व्हाट यू कॉल दैट? जिशान सर, आपको पता है क्या बोलते हैं वो जो कारों पे लगा देते हैं?
सक्शन कप होता है सर वो।
नो आइडिया सर।
अच्छा चलिए, मैं अभी क्या सक्शन पंप की तरह भी बोल सकते हैं, अदरवाइज let me search it, but first let it finish. I will show you that structure, what is that, and I will show you the importance of this. So this is a structure like this. सनलाइट प्रोटेक्टर है, उनके बीच में रबर की वो होती है. I will show you, and the function is same in both the cases. I tried to show you from the different point. Okay, so see, this is one more, and the more enlarged view. This is the same structure which you have already seen. This is the still photograph of the same slide. Actually, we were trying to detach out the coleochete from a wooden block. Okay, but along with coleochete, we also found all these young thallus, young filament of Oedogonium. This is one more structure. You can see there are some fiber-like structure. So it may be anything. It may be a young Oedogonium, or it may be a part of the tissue. Here you can see there are so many structures. So it may be a part of that angiospermic thallus, जैसा निकला है, because it's not taking any stain of iodine. Yes, let me search it in Google. I'm just stopping my screen sharing for some time. Once it's there, I will show you. Is it visible to everyone? ये आपने देखा है?
Yes sir.
Okay. In the center of this, there is a rubber-like structure. So
Sir,
when
vacuum
you place
stickers. Vacuum stickers.
Yeah, vacuum. Thank you, dear. Thank you. So I will type vacuum sticker. Thank you, dear. I was not able to. Yes, this is the structure. Okay. This is actually resembling with the. किसे resemble करता है ये? Hold fast से. And what is the function of this vacuum sticker? क्या function होता है?
So for attachment.
For attachment, and the same way, hold fast get attached itself with the substratum. अदरवाइज कितना लंबा फिलामेंट है? 30, 40, 50, 60, 100 of cells long फिलामेंट है. So how it will remain attached with the substratum? It need a firm binding or firm attachment with the substratum, and that binding will be provided with this kind of structure. So I hope now you can resemble this structure with that structure. Can you? कर सकते हो ना?
Yes, sir.
Okay. चलिए. So that's the same thing I want to explain. The next is your apical cell. हमने bottom की बात कर ली. Now we are discussing the apical, means the topmost part. And the free end at the upper part of the filament is broadly rounded, forming a dome-shaped apical cell. And this is the first time when we were able to see the apical cell. इससे पहले हमने जितनी बार भी slides बनाई है, we were not able to find it out. That's why we made this. Yeah, this is the apical cell. Can you see it? Only focus right now on the topmost cell. It's a dome structure. Okay? This is this one. These are the apical cells of Oedogonium. This one as well. Now the third part is the intercalary cells. Okay? Cells between the basal and apical cells are intercalary cells. जो होल्ड फास्ट के बीच में और डोम शेप्ड एपिकल सेल्स के बीच में जितनी भी सेल्स हैं, दे आर नोन एज intercalary cells. And the distinctive feature of Oedogonium is the presence of characteristic ring at the apical end of some intercalary cells. You can see this is the structure. I will enlarge this portion. Can you see?
Yes, sir.
This ring-like structure. This is your ring-like structure or caps. And the cells which contain this cap is known as cap cells. Okay. These rings are formed at the time of cell division and are called apical ring or apical caps. Only those cells which have the apical ring divide again, and the cells that have undergone many such division display several rings. So by just counting these rings, you can calculate how many times these cells have been divided. Like this, 1, 2, 3, 4. 4 ring I can see here. So we can assume that these cells have been divided 4 times. Okay. There is only one ring visible. So it may be possible that it's divided only one time. So depending upon how many times it has been divided, the ring formation will be the same number of as per the division. These are some more photographs. Here, please focus only to that structure जो हम दिखा रहे हैं. There are so many other structures also. We will discuss it later on. Okay, you will see the same photograph in the later half of this video or later half of this lecture, and at that time we will focus on this structure. Right now we are focusing only on this structure. जैसे अर्जुन को एक टाइम पे चिड़िया की आंखें दिखी थी, इसी तरीके से आपको भी फोकस करना एट ए टाइम ओनली दिस स्ट्रक्चर. वी आर फोकसिंग ऑन these cap cells. Forget about this structure and forget about this structure. We are not bothered about it right now. We will discuss this structure later on. Here is one more such kind of structure. Okay. So अभी तक जो हमने कर लिया है उसके हिसाब से what you have to do in practical. First of all, you have to make a filament. Okay, you have to make a filament. But sometimes students make filament like this. Direct filament बना देंगे. But you should make this structure like that. Can anyone explain what Does it meaning when we are joining the two part of the filament by dotted line?
Sir, it's not completed. It's in continuation.
Yeah, it's in continuation. There are many more cells in between this cell and this cell. Okay, so the meaning is very clear. Okay, you have seen that some of the cells are having cap cells, so you can make some cap cells. Okay, otherwise you can make normal cells. There are some cells which are having cap. There are some cells which are don't having any caps. This is the way you will make the holdfast, but it's only a diagrammatic representation. When we draw it, and all the drawings are done by the Yani ma'am. So when she draw this structure, she got a photograph like this. So now you have a different photograph. You have observed different things in the previous slide. So you have to draw accordingly. So what you have to do here, just try to take a screenshot. Okay, I will share this entire video with you all, and take the printout of the hold fast and draw the same hold fast. जैसा आपको hold fast दिख रहा है, like just if you are taking the printout of this, so you have to draw, paste it and draw it as it is. Okay, these structures are only for your help. Okay, how simply you can make. Okay, is it clear?
Yes, sir.
Yes, sir.
And this is a vegetative cap cell. This is also one of the drawing. So you can also take printout of some of the screenshot from this video, and whatever you are taking out, screenshot are you taking out, you can paste it in black and white or color, whatever suit you, and try to draw in the same manner. This is only for your guidance, okay, how we can draw it. जैसे अभी आपको पीछे की, suppose you want to draw it, but how you will draw it? There is no need to draw all these dotted structure, because we are focusing only on these cap cells. Okay, if you want to show the vegetative cell as well along with the cap cell, then you have to make these internal structure of the cell as well, which we will discuss later on. Okay, so till now we have discussed what is the morphology, what are the three different kind of cells. Okay, the same way you can also make the apical sac. Earlier we don't have any photograph of apical cell, but right now we have. So you can also draw first a single filament of Oedogonium, hold fast of Oedogonium, apical portion of Oedogonium, a intercalary vegetative cap cell. Okay, so these will be the four diagrams you have to draw till now. जितना हमने डिस्कशन अब तक किया उसमें 3-4 डायग्राम आपके हो गए हैं. Then we come to the cell structure. The cells of the vegetative filament are elongated. You all have seen that, and cylindrical with a rigid cell wall including the protoplast. The cell wall, except that of holdfast, is differentiated into 3 layers: outer chitin, middle of pectin, and inner cellulose layer. So this point is for your theory part. Okay, no need to mention this particular point in your practical, because you cannot see whether it's made up of cellular or not. Okay, is this point clear? Whenever
Yes,
you are
sir.
writing in your practical record, please mention only those points which are actually visible. जो आपको दिखता है केवल वही मेंशन करना है. No need to try to copy it from the slide, from the notes, or from the videos. This is for your theoretical knowledge. It's not for your practical knowledge. Okay, yeah, this thing we can see. The plasma membrane encloses the cytoplasm, a single large chloroplast with a number of pyrenoids. I will show you in the coming slides. A single parietal nucleus, मुझे नहीं पता कि दिखेगा नहीं दिखेगा, and other membrane bound organelles. such as mitochondria, Golgi body and endoplasmic reticulum. So whatever things is visible in your slide, you have to mention only that. The rest of the things is keep it aside for your theory exam. And the chloroplast is large, reticulate structure made up of hollow cylindrical network with message, and the chloroplast is parietal in position and extends from one cell to another. And the chloroplast of Oedogonium contain microtubule which provide support to the large chloroplast. Apart from this reticulate structure, no other things you can observe. So just mention only this. There is a large chloroplast which is reticulate in structure. That's it. Finish your comment up to this point in your practical record. But it is only for practical, not for theory. And there is a large central vacuole filled with the cell shape and a series of transverse parallel fragile rings that we have already discussed. What is this cap cell? So here you can see this is a intercalary cell and enlarge view. Now we will focus only in this internal structure. So we enlarge it. So can you see this dotted structure? This one?
Yes, sir.
Yes, sir.
Okay. So these rounded structures which are comparatively more darker than the rest of the structure are pyrenoids. And you can see that there is a net-like structure of chloroplast. Means there is no definite structure. It's a net-like structure and it is throughout the cell. So that's what is known as reticulate chloroplast. Okay? These dotted-like structures are pyrenoids. Okay, so this is the diagrammatic view, but I will not suggest you to draw it in this way. Please draw it in the same way as we have shown you here, because you are observing structure like this. Okay, आपको इस तरह की structure दिख रही है where you will see a net-like structure, in between there are dotted structure. Am I right? यही दिख रहा है ना आपको?
Yes, sir.
So please try to draw only that which is visible under your microscope. And all those photographs which I have shown you are taken by us or our student. आपने या तो आप जैसे आपके जो सीनियर्स हैं उन्होंने फोटोग्राफ्स लिए, ये ज्यादातर फोटोग्राफ्स हम लोगों ने लिए. So there is no copyright issue. You can paste it anywhere where you want. Okay, but please don't use this. It's only just to show how you will label it. Now comes to reproduction. In Oedogonium, reproduction takes place by vegetative, asexual and sexual method. Vegetative reproduction is generally takes place by fragmentation and akinete. Fragmentation may take place by dying out of some intercalary cells. Means it may possible that it's a long filament and in between there are certain cells which are start dying out. अब just imagine कीजिए you have a long thread like structure और बीच में से उसमें it become very delicate. So what will happen? That the rope or that thread will divide into different fragments. Yes or no?
Yes, sir.
Okay. Suppose this is a long filament. Here there is a hold fast. So this cell may become delicate or start dying out. This cell, this cell, this cell. So what will happen? It will break down into this fragment, this fragment, this fragment, this fragment, and this fragment. So it may possible some of the fragment will die out because of the unavailability of the proper nutrition, or if they are strong enough cells, so they will start developing the apical cells or the holdfast cell or both. Okay, and they will start dividing from them. So each and every fragment may be able to develop into a complete filament. or it may be possible because of accidental breaking of the filament, and it's a long filament, it may get break, and each and every part can develop into a complete filament. Or it may be possible that it's an adverse environmental condition. Okay. So there are so many things which can divide the complete filament in different parts. And in some species, vegetative akinets are developed during unfavorable conditions. Apart from that, there is asexual reproduction is also take place, and the main phenomena is stephanocontian type of zoospore. Now, कौन बताएगा मुझे? Kanishka. Yes, Kanishka, you will tell me what is the meaning of stephanocontian type zoospore. Is there anyone who can tell me what the meaning of stephanocontian type? Otherwise, we will mark a complete absent for the entire class. Bhupesh Nina. Yes, Bhupesh.
Yes, sir. Staphylocontin type means numerous short flagella in the subapical portion.
Yes, that's really great. So stephanocontin type means zoospore with numerous short flagella. Normally, we can see one flagella or two flagella in Chlamydomonas. Okay, but in this particular genus of your syllabus, you will see whatever is the motile structure, almost all of them are contain numerous short flagella, and that structure which contain numerous short flagella is known as stephanocontin type. Okay, the cap cell become a zoosporangium because the cells which have the ability of division can be developed into a zoosporangium. And how it will develop? It will start accumulating the storage food, and a single cell will become a zoosporangium, and zoospore will take place or will get produced singly, means inside a zoosporangium only one zoospore will take place. Here you can see the scanning electron micrograph of zoospore. These photographs are not taken by us. That's why we gave the proper credit from where we have taken these photographs. So you can see, even you cannot count the number of flagella emerging out from this apical portion of this zoospore. Okay. So it's an artistic work after taking the scanning electron micrograph. It will give you black and white portion only. Just to give you a better appearance, someone stain it with yellow color, and here the entire background has been removed. So it will be more clear to you about the structure of a zoospore. But in practical, what you will write? You will simply write that asexual reproduction takes place by zoospore, which is of stephanocontin type, and in bracket you will write multiple flagellated zoospore. But these are all part which you will write, remember, only for your theory purpose. At the time of zoospore formation, the entire protoplast of zoosporangium withdraw from the cell wall as a single unit. जितना भी प्रोटोप्लास्ट उस सेल्स के अंदर था, which will develop into zoosporangium, will become a single unit of the entire cell, and its nucleus retracts somewhat and move towards one side. The entire protoplast round up, and then on one side a hyaline region. Please note down this hyaline region. Hyaline region appear adjacent to the nucleus, in between the cell wall and nucleus. Along the margin of this hyaline region, a circular ring of blepharoplast. Blepharoplast पता है आपको क्या होता है? उसे ही बताया था मैंने आपको ओडोगोनियम में, सॉरी क्लेमाइडोमोनास में.
Yes, sir. Basal body.
A basal body, especially
basal
of a flagellated cell.
Yes. So it will, these are those granules from which individual flagella will be take place. So there will be approximately 30 blepharoplasts, which will give rise to 30 short flagella, and that's why that you will see a multiflagellated zoospore. So earlier, when it was inside a nucleus, it was a rounded structure, and nucleus will move towards this side, and on this portion there is the appearance of blepharoplast, and those granules will give rise to these multiple flagella. And once I will show you it in this photograph, next coming photograph. Yeah. So this is the structure that this was a normal cell, and what happened in this? It will the filament start accumulating the storage material, and it become a vessel-like structure. And inside this, the entire chloroplast become rounded. And here you can see the one part, toward this part, it will start developing some blepharoplast granules, and from each blepharoplast granules, individual single short flagella will start appearing, and once it will come out from this structure. And how it will come out? These are the original photographs from this side. So this will come out in this way. It will break the cell and it will come out. And you can see flagella is still invisible. You can just assume because of there is some disturbance in the water, and with the help of this flagella, it will get attached itself with a substratum, and the moment it gets attached, it will release its flagella and start developing the new filament. So this photograph, diagrammatic representation, we have taken from Lee, and this is the original photograph we have taken from this site. Okay. In case there are some times it may possible instead of zoospore, aplanospore may also form. And what is the difference between the aplanospore और zoospore? क्या difference होता है दोनों में?
Sir, it's non-motile aplanospore and the other one is motile.
Motile, yes. The aplanospore don't have any flagella. I mean, the rest of the things will remain same. Their development will remain same. Their release will remain same. But apart from the flagella, the rest of the things, rest of the phenomena is same. It's aplanospore. Okay? चलिए अब हम बात करते हैं सेक्सुअल रिप्रोडक्शन की, विच इज मोस्ट इंपोर्टेंट इन दिस. Please ask if anyone have any doubt till now before we start the sexual reproduction. बताइए किसी को डाउट है कुछ? नहीं. चलिए. So sexual reproduction is an advanced oogamous type. What is the meaning of oogamous type? Oogamous.
Sir, female gamete is larger than male gamete. And one is motile and one is sterile.
Yes. Yes. So large, non-motile, male gamete. Large, non-motile female gamete, small, motile, male gamete. This is the basic difference between the oogamous type. Okay. Anisogamous, when one gamete is large, other is small, but both are motile. So that is known as anisogamous type. And isogamous, when both are motile and both are of same size. Why it is known as advanced oogamous type? Because not only the oogamous is bigger, non-motile, but also the male gamete may develop in different manner, macrantous type or nanantous type. That's why it is known as advanced oogamous type. There are distinct male and female gametes. The sexual gametes are formed in gametangia. Okay, male gametangia is known as antheridia and female gamete is known as oogonium. Okay, antheridium and oogonium, that I already told you. And sexual reproduction may be of macrantous or nanantous type. This point I added, but this point is only for your theory purpose. In many species of Oedogonium, it is relatively easy to induce sexual reproduction by placing old filament in fresh media and saturating the atmosphere with carbon dioxide. First, we will discuss about the oogonia, female reproductive structure. Oogonia are born on normal filament and developed by terminal or intercalary oogonian mother cell, which divide transversely in two daughter cells, upper oogonium and the supporting cell, supplementary cells. Okay, and it is not visible here. I will show you in the coming slide. Means one cell will divide, and it will divide. The upper portion will develop the oogonium, and the basal cells will develop into a supplementary cell. If one cell become another oogonial mother cell, then the oogonia are produced in series. Like here you can see it was a single filament, it got breakdown, but they were formed in a series. Oogonia contain a uninucleate, large, non-motile, green spherical egg cell with centrally placed nucleus. This is. But why it's not green over here? Why it's appearing like black or brown? Why it is so? Which stain we used?
Iodine.
Iodine. So it is possible that the starch is inside this egg cell, react with that iodine and become blackish in color, and the overall appearance is become brownish black. There is one possibility. There is one more possibility that it's no more an egg cell. It become a fertilized egg. That fertilization has been already taken place. So at that time, the other pigment will also start appearing like carotenoids, and then it will start appearing automatically brown color. तो दोनों चीजें हो सकती हैं कि या तो हो सकता है because of the iodine stain, or it may possible because the reproduction fertilization has been already taken place. But we cannot differentiate it whether it's a zygote or a unfertilized egg cell. Like in the zoospore, this egg cell, one part will become hyaline or receptive spot. Okay, I will show you in the coming diagram. It's not visible over here, and that is the site from where the male sperm or gamete will enter inside the cell. And oogonium has one or more distinct caps on its upper end. एक तो दिखेगी यहाँ पे हम नहीं विजिबल हो रही है अच्छे से, but yet it may have more than one as well. एक तो होगी ही होगी because it will develop in oogonium and supplementary cells. So एक तो confirm होगी, एक से ज्यादा भी हो सकती है. So here you can see there are a series of oogonia. Can you see all these structures? I will show in some high power. It will take some time in focusing. Yes. Can you see the structure? The rounded structure? Is it visible to everyone?
Yes, sir.
These are
Yes, sir.
oogonia. These are bigger than the normal cell. That's why it's a reproductive structure, and that's why the genus was named as Oedogonium. These are some still photographs. These are all original photographs. Here you can see multiple oogonia in the single filament, or here are single oogonia. Both type of filament may be there. This is one more photograph. Although it was not a good slide, but yes, oogonia was very clear. That's why we use this here as well. Okay. So how you will draw this oogonia? Just simply draw simple cells, rectangular cell, and then a ovum. And the bottom cell is known as supporting cell. Okay. There is some slight mistake in supporting cell. It should be either equal size of vegetative cell or should be smaller than vegetative cell. The next or male reproductive structure is known as antheridia. The filament is classified into two types of basis of the structure and position of antheridia. And the one is macranthrous type. The antheridia are born in the filament of the normal size. जो भी filament है उस पे develop हो जाए. And if the macranthrous monoecious species Oedogonia or antheridia develop on the same filament, अगर दोनों Oedogonia भी मिल रहा है, antheridia भी मिल रहा है, then it will known as homothallic or bisexual. And if they are in some species, if male reproductive structure, that is antheridia, is developing on a different filament and female reproductive structure, that is Oedogonia, is developing on a different filament, then it will known as heterothallic or unisexual. Okay? And the antheridia are produced in any capsule, just simply like it should be a capsule. The moment for developing any reproductive structure, whether it is Oedogonia or antheridia, the cell need to be divided. Okay? In Oedogonia development, there is only one division. Upper half will develop into Oedogonia, lower half develop into suppository cell. Whereas in case of antheridia, it will divide again and again. And the cell which will divide again and again is known as antheridial mother cell. And after every division, it will produce a chain of 2 to 40 uninucleate antheridia. Okay, I will show you. Here you can see small cells. These are this structure, this structure, this structure, this structure. All these are example of macranthrous species. Can you see these? These are no other than normal. And you can see here, yes, this is a very good photograph. I missed it. Can you see how many structure you can see inside a single cell? थोड़ा enlarge करके देख सकते हैं आप. कितनी दिख रही है यहाँ पे आपको? Two. Yes. So after for taking place of this chain of antheridial cell, there is a division inside the nucleus, and they will produce two male gametes inside a single antheridia. So they will produce 2 to 40 uninucleate antheridia, and those first we'll discuss the theory. And those antheridia will divide mitotically into two nuclei which form two anthrozoids or spur. And again, anthrozoids are unicellular, uninucleate and multiflagellate. Again I said, you have to remember, whatever motile structure it will take place in Oedogonium, it will be multiflagellate structure. Okay, it's a unicellular, uninucleate, multiflagellate, and which resembles zoospore. ये भी zoospore की तरह होती है, but अंतर क्या होता है? ये zoospore से छोटे, they are smaller than zoospore. Only the size will determine whether this is an anthrozoids or a zoospore. And they are yellowish color because of the reduced plastid. I will show you in our class, photograph as well. And the anthrozoids liberate same way as zoospore. जैसे zoospore cells को break करके निकला था, ऐसे ही हमारे anthrozoids will also come out after breaking the cell by transverse splitting of the wall of antheridium, and then it will get attached or it will move towards the female gamete. Like this. तो आपको मैं दिखा देता हूँ ये. So here you can see this structure. The inner part is slightly yellow. It's not taking the same amount of color as the rest of the cells. Am I right? ऐसा है या नहीं है?
Yes, sir.
It's because कि हमने अभी पढ़ा है कि they have reduced chloroplast, and reduced chloroplast means low amount of starch will be there compared to the rest of the cells. Here it's a young antheridia because there is a no division of male nuclei. यहाँ पर division नहीं हुआ है, यहाँ पर division हुआ है. Just you have to note down this point. There are, you can see two nuclei. यहाँ पे we can see only one nuclei. Here we can see two nuclei, but here we can see only one nuclei. So one nuclei means it's a young male gamete. Two nuclei means that mitotic division has been already taken place. Here also one, in this as well as one. Here we can see two. In this photograph, it's a permanent slide photograph. We can see only one nuclei is there. Here also one nuclei is there. So you have to make sure when you are drawing the Oedogonium macrandus male filament, if you are pasting a photograph which is showing two nuclei, you should draw two nuclei inside the antheridium. If it is showing one nuclei, you should show one nuclei. तो ऐसा नहीं कि दो गलत है या एक गलत है. Both are correct. It depends upon what kind of at what stage you have observed this macrandus male filament. Okay. Next, the last point is nanandrous type, and what it is, the antheridia are produced on especially very small 2 to 4 cell filament, which is known as dwarf male and nanandria. How this nanandria will take place? These dwarf male grow epiphytically attached to the female filament, hence nanandrous species are always heterothallic or dioecious. अब देखिए हेटेरोथैलिक क्या होता है, जब when male and female gametes will develop on two different filaments, and here it is an essential part that male gamete or male will be produced by the dwarf male, and dwarf male is a separate filament, so they are always heterothallic or dioecious in nature. A dwarf male is produced by a spore called androspore, and the development of androspore takes place within androsporangia. The development is almost same like your zoospore, like your anthrozoid formation. The only difference between androspore, anthrozoids and zoospore, please note it down right now. Zoospore, the biggest one, androspore, the second biggest one, and anthrozoids are the smallest one. Rest of the structure will remain same. If you will see a anthrozoid, a zoospore, or an androspore, all the structure will appear similar. Only based on the cell size, you can differentiate them. And if the nanandrous species of the androsporangia and oogonia are born on the same filament, जिस स्पोर से ड्वार्फ मेल बनेगा, अगर वे सेम फिलामेंट में बन रहे हैं, मीन्स oogonia भी वहीं पर है और nanandria का स्पोर भी यानी एंड्रोस्पोर भी वहीं पर है, then that filament is known as gynandrosporous, and species known as gynandrosporous species. And if this androspore is developing on different filament, then it is known as Oedogonium. Okay. Okay, this thing I already explained that zoospore are similar to androspore or anthrozoids. This has been already explained. So I will show you what happened. Yes. This is a slide. Here you can see oogonia is there, and I will show you. Yeah. Here, oogonia is there. On the bottom, there is cephalotory cell, slightly bigger than the other. Okay, this here. So what happens? Androspore will start germinate over here. इसके ऊपर develop हो जाएंगे. So you can see 2 cells is there. There may be more than 2 cells, 4 cells, and they will always get attached near to this oogonia. This is oogonia, and they will get attached with its cephalotory cell near this oogonia. And inside this dwarf male, the anthrozoids will develop, or antheridia will develop, and they will swim and will get attached with this or will penetrate this oogonial wall and get mixed the male gamete with the female gamete. So you can take a screenshot of this portion as well, so that it will be easier for you. Or we have already discussed all these things. So this is the structure of, this is a drawing of Nanandria. Okay, this is again, I'm saying it's a diagrammatic view. Whatever you are observing, you can draw that, but you can take help how to draw it. Fertilization, take a screenshot of this. I will explain in the photograph. So whether it is dwarf male or whether it is inside a macrandus species, once the male gamete will come out from the antheridia, it will swim towards the female gamete or oogonia. And this is a hyaline sign. We explained there is a region, hyaline region or receptive region, from which this will get slightly break and it will allow the entry of this male gamete. Okay. The moment it will enter inside this oogonia, it will release its flagella and get mixed the male protoplast with the female protoplast, and they will form a diploid zygote. So this is about the fertilization. And zygote liberate by the decay of oogonial wall. So this, inside this structure, zygote may remain up to 1 year. Means if there is no sufficient water, no nutrient is there, so a viable zygote can remain inside this structure protected from the other environmental things. And once it gets liberated again, in prior to germination, diploid nucleus of the zygote undergoes meiotic division to form haploid nuclei. So what kind of life cycle it will have? Where the zygote is only diploid structure, it is in haplontic type. Earlier it was haploid. After before liberation, it will become haploid, and only the zygote is the only diploid structure. So it's a haplontic type of life cycle. Okay, it will divide into 4 daughter cells, and each and all daughter cells will come out in the form of meiozospore, and meiozospore are liberated by rupturing the zygote wall and remain enclosed in a vesicle which soon disappear, and whenever they find it suitable substratum, they will get attached on that substratum and will produce the new filament or new act as a multiflagellated meiozospore or zoospore. तो ये उनका पूरा जर्मिनेशन है कि एक बार जाइगोट बन गया तो that first it will have to undergo in a meiotic division to produce 4 daughter cells, and then all those daughter cells can come out in the multiflagellated. I told you in the beginning, whether it is zoospore, whether it's enterospore, whether it's anthrozoids, or whether it's meiozospore. Why we are knowing it's meiozospore? Because it's a result of sexual reproduction. It is taking place because of meiotic division, and it appears like a zoospore. That's why it is known as meiozospore, multiflagellated meiozospore. So this is all about the life cycle of Oedogonium. What can happen? This had been, we have already discussed how the zoospore will come out and germinate. Here you can see this is a oogonia. Okay, it may possible that some of the cells will produce enterospore. Enterospore will germinate near the oogonia, and then this enterospore will produce anthrozoids. It will get fertilized, will form zygote, undergo meiosis, will form 4 daughter nuclei. All the nuclei will come out as a meiozospore, and then they will germinate in the new filament. दूसरा तरीका और भी हो सकता है कि this is the oogonia, and instead of producing this dwarf male, there is one more filament. I will show you here. There is one more filament which is having anthrozoids, and these anthrozoids will come out and get fertilized with this, and ultimately they will form zygote. So instead of taking this place of dwarf male, they can take the direct route of this. So this is the life cycle of, there are two ways of life cycle, dwarf male's life cycle and the normal macrandus type life cycle. And this is a classification proposed by Lee with justification. Up to chlorophytes we have already discussed in case of Chlamydomonas and volvox. We will discuss Oedogonians, which have only single family Oedogoniaceae. And why it is under Oedogoniaceae? Because of uninucleus filamentous freshwater alga with a unique type of cell division. This is the most important thing. Unique type of cell division, motile spore, world of flagella. And these are the characteristic features which makes any genus which will come under Oedogoniaceae. And there are only 3 genus. Oedogonium is the unbranched, whereas 2 others are branched. And you can also again differentiate between the other 2, Oedocladium and bulbocute. One is having with long colorless hair, bulbocute, and the other is not having any hair. तो ये आपके 3 जीनस हैं इस फैमिली के. तो वी कैन जस्टिफाई अप टू Oedogonium. So you can take a screenshot of this as well. This is the way we used to teach in our class. And I am showing you one more additional information. When and where nanentday will take place, it's not required neither for theory nor for practical. But take a screenshot, you will find it really interesting when and where the endospore will take place. So that's it from my side for Oedogonium.